Children, last class I have completed female reproductive system of human, and hope you are thorough with us and clear with us also. And today I am going to start gametogenesis, process by which the gametes are formed. Okay, so what are the gametes? Children, you know very well what are gametes. Yes, there are two types of gametes, that is a male gamete and female gamete. Gamete is nothing but the sex cells, that is sperm in male and ovum in female, or the gametes. And how the gametes are formed in the male and female reproductive system, right? So you know children, in our body there are two types of cell. One is somatic cell, another one is sex cells. The somatic cells are diploid in nature, that the chromosome number are in two sets. This 40, say for example in human the somatic cells consist of 46 chromosome, that is 23 pairs. But in sex cells such as ovum and sperm have only a single set of chromosome, that is 23 chromosomes alone. Okay, so how this chromosomes are reduced, how the number of chromosomes are reduced in the sex cells. So it is taken place by a process called gametogenesis, and also the production and maturation also of the gametes also taken place by the process of gametogenesis, right? Okay. So how the number of chromosomes are reduced in the sex cell is, it is by the process of cell division called meiosis. Meiosis is called a reduction division. Why it is called so? Because the number of chromosomes are reduced by the meiosis cell division. But in mitosis cell division, the number of chromosomes will not be reduced. The number of chromosomes will be as such in the parent cell. But in meiosis, the number of chromosomes are reduced into half. So by the process of meiosis, the gametes are acquiring the haploid set of chromosome. And next question, why should the gametes have only haploid set of chromosome? Okay, so the is only male, only sperm in male and the ovum in female or the sex cell. Those cells alone have haploid chromosomes. Other cells, they are having diploid chromosomes only. So why there will be a haploid number of chromosomes in sex cells? Not only in human, in all sexually reproducing animals, they are having only haploid number of chromosome in their sex cells. Okay. So why should they maintain the haploid nature of chromosome is that you know very well children, the number of chromosome in each species is constant. The number of chromosome is constant in each species, isn't it? Say for example human, the number of chromosome in all the cells will be how much? 46. If you take any person or any place in any part of the world, if you take all the persons of human having only 46 chromosome in their body cells, in their somatic cells or body cells. So this should be maintained by the sex cells only. Why? Because see the sex cells, how the young ones are produced, you know very well. By the fusion of male and female gamete, the young ones are produced. Okay, when the sperm and ovum fuse to form a zygote, when a sperm and ovum fuse to form a zygote. The zygote is a single cell nature, and it has diploid chromosome now. How it became a diploid chromosome is it? See, the sperm has 23 chromosome and the ovum has 23 chromosome. When both the nucleus fuse together to form a single nucleus, when they fuse together, the nucleus also, nucleus fused together, the chromosome inside the nucleus also fused. So 23 plus 23, and the zygote has a 46 chromosome. So what happened very next? The zygote immediately undergo mitotic cell division. You know very well what is mitotic cell division. Mitotic cell division is nothing but one cell divide into two with the same number of chromosome as their parents. Okay. So when the zygote undergoes cell division, it divide into two cells. Both the cells have 46 chromosome. So like that, the zygote undergoes cell division, form a embryo develop into young one. So what the number of chromosome is present in the zygote will have the particular young one developed from that zygote also, right? So that to maintain a constant number of chromosome in the future progeny, the haploid set of chromosomes are maintained in the sex cells, right children? Okay, let's enter into the topic. So gametogenesis, I said the process of formation of gametes in male and female is called gametogenesis. Spermatogenesis. What is spermatogenesis? The process of formation of male gamete, that is sperm, is called spermatogenesis. The same way, oogenesis. What is mean by oogenesis? The process of formation of ovum in the female body, that is in the ovary, is called what? Oogenesis. Okay. So in this topic, we are going to see how the sperms are developed by the subsequent stages, okay, and oogenesis, how the ovums are developed, all we are going to discuss now, okay. And the difference between gametogenesis, oogenesis, that you can find from the diagram itself, okay children. Gametogenesis is the process of formation of gametes, okay, that is sperm and ovary, sperm and ova from, not ovary, ova. It's a mistake given here children. Sperms and ova from the primary sex organs in all sexually reproducing organism. What are the primary sex organs? Testis in male, ovary in female are the primary sex organs. Sperms are produced in the testis, ova produced from the ovary. And then meiosis plays the most significant role in the process of gametogenesis. As I said, which cell division play a significant role in gametogenesis? Meiosis. Okay. So let's enter into the topic spermatogenesis. Spermatogenesis is the sequence of events in the seminiferous tubules. Okay. Already I said when I taught what is male reproductive system, seminiferous tubules only sperm production takes place, and maturation, till maturation the sperms are stayed in the seminiferous tubules only, that is the Sertoli cells nurture the sperm, developing sperm அப்படின்னு சொல்லியிருக்கேன் நான் உங்களுக்கு. Spermatogenesis is the sequence of events in the seminiferous tubule. So spermatogenesis taken place in the seminiferous tubules of the testis that produce the male gametes, the sperms. During development, the primordial germ cells migrate into the testis and become immature germ cells called sperm mother cell or spermatogonia in the inner surface of the seminiferous tubule. The spermatogonia begins to undergo mitotic division at puberty and continue throughout the life. Okay children, as per this paragraph, what they are trying to say, that a spermatogenesis is a sequence of events. It taken place in the seminiferous tubules of the testis and produce the male gametes called testis. Sperm. How the sperms are produced, from which cell act as a sperm mother cell, okay, that is primordial germ cells migrate into the testis. So primordial germ cell, it is migrate into the testis and become immature germ cells or spermatogonia in the inner surface of the seminiferous tubules. Then the spermatogonia begins to undergo, okay, that is primordial germ cell which has migrate into the testis and become the immature germ cell called germ mother cell. It is otherwise called spermatogonia. So these are taken place in the inner surface of the seminiferous tubules. Next, okay, this immature primordial germ cells become immature sperm mother cell or spermatogonia. Next, the spermatogonia undergo mitotic division at puberty and continue throughout the life. This you should note down children. Spermatogonia undergo mitotic cell division and produce primary spermatocyte children. Okay, see here, you can observe in this figure. You see spermatogonia which undergo mitotic cell division and produce primary spermatocyte. Okay, this primary spermatocyte undergo meiosis 1 and produce 2 secondary spermatocyte. This secondary spermatocyte undergo meiotic 2 division and form spermatid. Spermatid undergo spermiogenesis and form the spermatozoa. With this schematic representation itself, we can understood there are 3 cell division taken place in the process of spermatogenesis. First cell division, spermatogonia become a primary spermatozoite, it undergo mitotic cell division. So it is understood spermatogonia is diploid in nature and primary spermatozoite also diploid in nature. The primary spermatozoite which undergo meiosis 1. Meiosis you know very well, it is a reduction division. So when the primary spermatozoite undergo meiosis 1, the chromosome number is reduced into half. You can observe in the diagram itself, children. Primary spermatozoite is 2n in nature, that is diploid in nature. Secondary spermatozoites are n in nature, that is what? Haploid in nature. Again the secondary spermatozoite undergo meiosis 2 to form a spermatid. That's all. The cell division will be stopped with that. After that what happened? The spermatid undergo a process called spermiogenesis, by which the spermatid converted into spermatozoa. That's all about spermatogenesis. So by one thing you should note, children, there should be a difference between spermatogenesis and oogenesis. Spermatogenesis and oogenesis. See, you compare in spermatogenesis, spermatogonia. In oogenesis, it is oogonia. So both spermatogonia and oogonia undergo mitotic division. And what is the next result? The oogonia become a primary oocyte. Here the spermatogonia become primary oocyte. Till that same only. Next, the primary oocyte undergo meiosis 1. Meiosis 1. Same way primary spermatozoite also undergo meiosis 1 and produce 2 secondary spermatocyte. How many secondary spermatocyte? 2 secondary spermatocyte. But the primary primary oocyte undergo meiotic cell division. When the meiotic cell division is taken place, completed prior to ovulation. Meiosis 1 completed prior to ovulation. Ovulation, what is ovulation? All we will discuss later. Okay. So what will be the what we want to discuss here? In spermatogenesis, primary spermatocyte undergo meiosis and produce 2 secondary spermatocyte. But in oogenesis, this is the first difference. Primary oocyte undergo meiotic cell division and will not produce 2 secondary oocyte. It will produce 1 secondary oocyte and 1 polar body. First polar body is formed during the meiotic 1 cell division. Okay. Okay. Leave it. And next one. See here. Secondary in spermatogenesis, secondary spermatocyte which undergo meiosis 2 and produce 2 secondary spermatocyte undergo meiosis 2 and produce 4 spermatid, and the 4 spermatid develop into 4 spermatozoa, that is sperm. But in oogenesis, the only 1 secondary oocyte which undergo meiotic cell division and produce only 1 ova and another one is second polar body. So what will be the result at the end of spermatogenesis? 1 spermatogonia produce 4 spermatid, but at the end of oogenesis, 1 oogonia produce only 1 ova. This is what a difference between spermatogenesis and oogenesis. If you clear with this diagram, children, you can write the difference between spermatogenesis and oogenesis. And you can write the details of process of spermatogenesis as well as detail of process of oogenesis. Okay, children. So let's enter into the text side, children. Already I completed what is that? Till the spermatogonia undergo mitotic division at puberty and continue throughout the life. Okay. What is the next? In the so you could clear with this idea, I think so. So I will read out and go. Suppose if you have any doubt, you can clarify with me. Okay, children. In the first stage of spermatogenesis, the spermatogonia migrate among the Sertoli cells towards the central lumen of the seminiferous tubule and become modified and enlarged to form a primary spermatozoite which are diploid with 23 pairs, that is 46 chromosome. Okay, in the first stage, that is spermatogenesis, what happens? Spermatogonia migrate among the Sertoli cells. You know children, seminiferous tubules, it is a tube-like structure. It is lined with the Sertoli cells and also it consists of a lumen. You know very well, wherever the tube is there should be a lumen inside. Lumen means what? Cavity inside that. So what the spermatogonia will do? The spermatogonia which move along the Sertoli cells and enter into the central lumen of the seminiferous tubules and become in the lumen of the seminiferous tubules, it become modified and enlarged to form the primary spermatozoite. And the nature of chromosome in primary spermatozoite are the 23 pairs, that is 46 chromosomes. Okay. Next children. Some of the primary spermatozoite undergo mitotic. Some of the, not all the primary spermatozoites. Some of the primary, okay, spermatogonia undergo mitotic cell division, produce primary spermatozoite. Okay, we will clear. Next, what is the next sequence of this spermatogenesis is some of the primary spermatozoite undergo first meiotic division to form two secondary spermatozoites which are haploid with 23 chromosomes each. The secondary spermatozoites undergo second meiotic division to produce 4 haploid spermatid. The spermatid are transferred into spermatozoa or sperm by the process is called spermiogenesis. These are called 2 Marcos in spermiogenesis. Sperms are finally released into the cavity of seminiferous tubule by a process called spermiation. So, sperms are finally released into the cavity of the seminiferous tubule and this process is called spermiation. Please bracket it, 2 Marcos in. The whole process of spermatogenesis takes place about how many days? 64 days. From spermatogonia to the formation of spermatozoa, it taken 24 days, 64 days. And the formed sperms are released into the cavity of seminiferous tubule by a process called as spermiation. At any given time, different region of seminiferous tubules contain spermatozoite in different stages of development. So at any time, the seminiferous tubules of the testis consist of different stages of development of spermatozoa. See, when we see the interior section of seminiferous tubule under microscope, we can see the various stages of spermatozoa. You can see primary spermatozoite, you can see spermatogonia, you can see secondary spermatozoite, you can see spermatid, all the stages you can see at a given time. And the sperm production remains nearly constant at a rate of, what is that? A constant rate of sperm production, 200 million sperms per day. Spermatogenesis starts at the age of puberty and is initiated due to the increase in the release of gonadotropin releasing hormone by the hypothalamus, that is GnRH, gonadotropin releasing hormone acts on the anterior pituitary gland and stimulate the secretion of 2 gonadotropins, namely follicle stimulating hormone and luteinizing hormone. FSH stimulate, that is follicle stimulating hormone, stimulate the testicular growth and enhance the production of androgen binding protein by the Sertoli cells and helps in the process of spermiogenesis. So, androgen binding protein is the protein produced by the Sertoli cells and then helps in the process of spermiogenesis. Next, luteinizing hormone, LH hormone, acts on the Leydig cells and stimulate the synthesis of testosterone, which in turn stimulate the process of spermatogenesis. There are two processes, one is spermiogenesis, another one is spermatogenesis. These are the two processes. So, you will have a question like discuss about the hormonal functions of spermatogenesis. Okay. So, or write the role of hormones and its function in the process of spermatogenesis, like that you will have. So what happened? First, at the age of puberty, the gonadotropin releasing hormone will be increased in the hypothalamus, right? This gonadotropin releasing hormone will stimulate the anterior pituitary hormone. This pituitary gland, this anterior pituitary gland release two gonadotropin hormones, namely follicle stimulating hormone and luteinizing hormone. What the follicle stimulating hormone will do? Follicle stimulating hormone stimulate the production of androgen binding protein by Sertoli cells. This androgen binding protein help for the process of spermiogenesis. You know, children, what is spermiogenesis? Next one, LH, luteinizing, which act on the Leydig cell. Leydig cell also present in the seminiferous tubules. This Leydig cell secrete the hormone testosterone. This testosterone stimulate the process of spermatogenesis. Okay, so that's what the role of hormone played in the female body to enhance the process of spermatogenesis. Next one is structure of spermatozoa. You will get as a 5-mark question, children. The human sperm as a microscopic flagellated and motile gamete. So, human sperm as a motile in nature. Human sperm as motile in nature, that it can swim, it can move, and it has flagella. Since it has flagella, it will move. The whole body of the sperm is enveloped by plasma membrane and is composed of it can head, neck and a tail. Head comprises of two parts, namely agrosome and nucleus. Agrosome is a small cap-like pointed structure present at the tip of the nucleus and is formed mainly from the Golgi body of the spermatid. So, when the spermatid is converted into spermatozoa, the Golgi bodies in the spermatid is modified into a agrosome. The agrosome is present in front of the head, that is above the nucleus region, and it contains an enzyme called hyaluronidase. The agrosome consists of an enzyme called what? Hyaluronidase. You can see the figure, you see, children. The spermatozoa whole body is covered with what? Plasma membrane, and body is divided into 3 regions: head, neck and tail. And the head is consists of 2 parts. One is nucleus, another one is acrosome. Acrosome is a modified form of Golgi bodies from the spermatid, and which is present in front of the nucleus. Okay, so please refer the diagram and learn the answer when you read this question. Okay. So, the agrosome consists of an enzyme called hyaluronidase. It is a proteolytic enzyme popularly known as sperm lysine. Underline it. Which enzyme is called as sperm lysine? Hyaluronidase. It helps to penetrate the ovum during fertilization. Hyaluronidase is an enzyme called proteolytic enzyme, otherwise called as sperm lysine, which help to penetrate the ovum during fertilization. How to help means, see, the egg is covered with a follicle. Egg is covered with 3 layers. Okay, zona pellucida, zona radiata and plasma membrane. So, this zona radiata are tightly held with the plasma membrane with a cementing substance called hyaluronic acid. So, the sperm cannot penetrate unless the hyaluronic acid penetrate the egg, unless the hyaluronic acid will be dissolved. So, once the sperm touch the egg, the agrosome release a hormone called hyaluronidase. The hyaluronidase dissolve the hyaluronic acid, the cementing substance hyaluronic acid, which tightly held with the egg, and loosen the cementing substance and make the way to enter the sperm into the egg. Okay, that's the function of hyaluronidase secreted by the agrosome. For then the nucleus is okay, next you come. What about nucleus? The nucleus is flat and oval, and then neck is very short and is present between the head and the middle piece. Okay, it contains that is, what is the part present, what are the parts present in the neck. It consists of proximal centriole towards the nucleus, which plays a role in the first division of the zygote. and distal centriole gives rise to the axial filament of the sperm. Okay, so neck region consists of only centriole, two centriole, proximal centriole, which is involved in the division of nucleus, first division of nucleus, and the proximal distal convolute tube gives rise to the axial filament of the sperm. The middle piece possesses mitochondria spirally twisted around the axial filament called mitochondrial spiral or Debetcan. Okay, so middle piece consists of a mitochondria which are in the twisted form, and this filament is called mitochondrial spiral or Debetcan. It produces energy in the form of ATP molecules for the movement of sperms. Okay. Next, the tail is the longest part of the sperm and is slender and tapering. It is formed of a central axial filament or axoneme and an outer protoplasmic sheath. Okay, so tail is the longest part of the sperm. It has a central part axoneme and is outer protoplasmic sheath. The lashing movement of the tail pushes the sperm forward. Human male ejaculate about 200 to 300 million sperm during coitus. It is estimated that around 60% of sperm must have normal shape of which at least 40% must show vigorous motility for normal fertility. Okay, so the sperm is the smallest human cell and the ovum or egg is the largest human cell. You note it, children. Next, see children, I plan to complete oogenesis today itself, but I couldn't complete already 26 minutes. Okay, I'll stop with this children. Homework, the structure of sperm and spermatogenesis, both. What the thing I taught today, both you will have as a homework children. Complete it then and there. You will have your quarterly examination by September 18th onwards, I think so. So the portion 4 lessons from zoology and 2 lessons from botany. Once I completed this lesson, I will start botany. So children, be concentrate and learn at home. Sit, don't waste your time. Prepare for your quarterly examination, right? Okay, I'll meet you in my next section with oogenesis. So complete your homework. Bye children.